Hello, everybody. This is Amanda Sierra from Azucarova's Center for Neuroscience. I think we are already recording this. Rosa Paulicelli is the other main organizer, and we have a fantastic team of people behind the scenes. Some of them will be working today. I can see Guy, who was the creator of this series. So thank you, Guy. Hello. Hi, Brown. He's there. So today we have as a speaker, we have Francesca Peri. She's a fantastic cell biologist who specializes in microglia. You will see terrific videos and images of live imaging of zebrafish microglia. I think she was actually the one who put zebrafish microglia in the map as a really interesting tool to study microglia behavior. And she's an expert in phagocytosis. My lab learned a lot from her research. I would also like to thank her in person because she's the first person who started the EMBL microglia meeting that we all love so much. So thank you, Francesca. That was a terrific effort. And now Rosa and I and other people are continuing with the effort. So now I'm just going to introduce Giorgio Corsi, who is a postdoc at Lausanne University, who will be introducing Francesca. And we also have Marta Pereira, who's a PhD student in my lab at Azucaro, who will be handling the questions and answers. So welcome, everybody, and your turn, Giorgio.
Thank you, Amanda. Good afternoon, everyone, and again, welcome to this second appointment of the European Microglia webinar series. Today, it is my pleasure to introduce to you Professor Francesca Peri. Francesca Peri is Italian, and she studied biology at the University of Padova in Italy. For her PhD, she moved to Cologne in Germany, where she worked with Siegfried Roth on the establishment of the dorsoventral and anteroposterior polarity in Drosophila. After obtaining her PhD in 2001, Professor Peri joined the lab of Christian Neuslein-Wohlacht at the Max Planck Institute for Developmental Biology in Tübingen, Germany. During this postdoc, she started working on how microglia engulf and digest dying neurons using zebrafish. In 2008, she became group leader at the European Molecular Biology Laboratory, also known as EMBL, in Heidelberg, where she continued working on microglia and their interaction with neurons using live imaging in the fish. Since 2018, she has been full professor at the University of Zurich. Today, she will give a talk entitled In the Belly of Microglia Towards a Dynamic Quantitative Understanding of How Microglia Phagocytose Neurons. Before we start, just a quick reminder about the questions. If you want to ask something to our speaker, you can either write your question in the chat, also during the talk, and at the end we will read it for you, or you can also just mention again in the chat that you have a question, and then you will be unmuted. And also, if you're willing to, we kindly ask you to leave the camera on so our speaker can see you during the talk. Thank you, and please, Professor Peri, the floor is all yours.
Thank you so much. Thank you, Giorgio, for this introduction. And I would really like to thank Rosa and Amanda for this invitation. This is a great honor, and also for taking over this fantastic seminar series. So thank you so much. So I'm gonna share my screen. Hopefully everything will go smoothly. Let me see. Okay. I hope you can see it. Yeah. And let me check that you can also see my pointer. Is it? Yeah, here. Do you see it?
All good.
Yeah, all good. Okay, as I said, thank you so much. Real pleasure. I'm going to start with something I'm sure many of you are very familiar with. And it's the fact that the fate of many cells in development and also homeostasis is actually death. Or to be more precise, is programmed cell death or apoptosis. And here you have some examples, how we separate our fingers by the death of cells in between the digits, the selection of T cells, turnover in the liver, in the intestine, photoreceptor outer-segment turnover, and so on. However, perhaps the best example of all is the development of the vertebrate nervous system. Because in vertebrates, what happens is that neurons are produced in large excess. And most of them will not be integrated into functional circuits, and they will undergo cell death. And the idea is that roughly half of the neurons that are produced in the vertebrate brain will undergo apoptosis. So this is a massive number of cells that die. Now, there is one important thing about cell death, and it's the fact that everything is controlled in the case of apoptosis. However, if the apoptotic cells are not collected, they are not removed, they will quickly undergo secondary necrosis. And this is, of course, a problem within a tissue because the content of the cell is leaking out, and this can lead to inflammation. And so effectively, the apoptotic cells are recognized and they are engulfed by professional phagocytes. And of course, I'm sure you know that the professional phagocytes, the best phagocytes in the brain, in the vertebrate brain, are actually the microglia. And the microglia are in charge of collecting, identifying and collecting dying neurons in two very important moments in the life of, I would say, a person, which is during development, during the formation of the brain, as I've just described, but also during neurodegeneration, because in many neurodegenerative contexts, Alzheimer, Parkinson, dementia, injury, stroke, and so on, these are characterized by the death of neurons, and it's the job of the microglia to find and remove these cells. Now, microglia, the interesting thing about microglia, when we compare microglia in development and neurodegeneration, and this is a study published a few years ago by the lab of the late Ben Barres, is that microglia in neurodegeneration and in development appears to upregulate the same genes. So these are either phagocytic factors or factors that are dealing with lipid metabolism. But there is really a high degree of overlap. And this actually tells us two things that are important. First of all, as I said, many of these are genes dealing with metabolism or trafficking of lipids. So this tells us that lipid metabolism and trafficking is important downstream of neuronal cell death in microglia. Many of these are actually also risk factors for Alzheimer, Parkinson, and so on. And then one other aspect it's very important here, and is the fact that since the same toolbox is basically genetic toolbox, is basically used in development and in pathology, then the development, the embryo, is actually a good place to study how microglia respond to neuronal cell death. So now, before I go into what we do and talk more about microglia, I would like to point out the fact that phagocytosis is actually a very ancient and very complex and well-orchestrated process. And there is a lot that we have learned from other cell types and from other model systems, but there is a lot still that we don't know about this process. So let me just say a few words. We can divide phagocytosis in roughly three steps. So the first one will be the recognition and the engulfment. The apoptotic cells, so in our case, these will be neurons, need to communicate their state to the microglia. This is through the release of long-range signaling, also short-range signals, so changes at the level of the plasma membrane. And in this way, the phagocytes, or in our case, the microglia, are able to identify the dying cell. After that, there is the process of engulfment. So the apoptotic cell is enclosed into a vesicle that is the phagosome, and the phagosome will undergo a maturation, mostly by fusing with other vesicle types, so lysosomes and endosomes. So effectively, at this point, the phagosome acquires the right enzymes and is able to digest the apoptotic cargo. And the third step and the last step is a point that that is clearly very important, but we know very little about this, and is the resolution. So what happened to the byproduct of digestion? So what happens to the lipids, what happens to the sugar, nucleotides, and so on? To which extent are these extruded, released in the extracellular space, or reused by the microglia, maybe to fuel their ability to actually engulf? So the crosstalk between resolution and the ability to engulf in the first place is very, very poorly understood. So I would like to conclude a little bit this introduction by asking, what can we learn when we study this process? So what is that is relevant for the microglia, but is very relevant in general for highly dynamic cells? So these are just a few questions that we have in the lab, and that to some extent I hope I can address today. So how do dynamic cells or microglia can navigate through complex tissue, in this case the brain, which is obviously a very complex, and the importance of this complexity, I'm sure, is clear. And how do microglia respond effectively to multiple and extrinsic and intrinsic stimuli? So multiple neurons dying, but also how their state, either health or pathology, impact on the ability to respond to these to stimuli that come from the outside. How do microglia adapt to changes? So for example, increases in cell death. And is there long short-range coupling between these phagocytes? So are microglia working alone, or do they really form a network? Are they able to compensate for each other? And then finally, something we know very little is what is the impact of apoptotic cell clearance on development and functionality of the brain, for example. So I spoke a lot about the ability of microglia to remove dying neurons. Let me give you some evidence that this is indeed something that is happening. This is the zebrafish brain, embryonic brain. I will tell you a little bit more about this model system in a second, but it doesn't matter. It could be a mouse, as Amanda has beautifully shown, or perhaps even the human brain. So what you see here are microglia in red. You can see this is one cell right here. And in green, you have acridine orange is a marker to that labels condensed chromatin, so apoptotic nuclei. You can see one here, another one here, another one here. So very few outside of the microglia, because indeed you can see that there is a lot of green material that co-localizes with the microglia. And in fact, if now we remove either chemically or genetically the microglia population, you see now that in the brain we find a lot of uncollected, or what they appear as uncollected neurons that obviously accumulate in this brain because of the absence of the microglia. So the question for us really for many years has been, how do microglia engulf these neurons? How they find them, how they can actually grab them, what happens to this material next, and so on and so forth. So as I said, we use the zebrafish mainly because of its optical transparency. So the embryo, the young zebrafish embryo, until 5, 6, and more days, is actually very poor in fat. So there is very little light scattering. And this is why you can go through and see cellular, subcellular processes very easily. So we can simply, label, we can simply, generate transgenic animals, where in this case, for example, the microglia are labeled with GFP that localizes at the membrane. And as you can see, these cells are highly dynamic and moving. So in order to better understand the interaction between the microglia and the neurons, and in particular the dying neurons, we have generated also a reporter for neuronal cell death. And this is based on the fact, you can see here in the schematic, that what happens to apoptotic cells, so one of the first things that happens in apoptotic cell is that phosphatidylserine lipids, which normally point towards the inside of the plasma membrane, this polarity is lost and they flip out. And so we made a transgenic animal where annexin 5, which is a protein that binds phosphatidylserine lipids with great affinity, is produced in neurons and is secreted, and is also coupled with the fluorophores. So effectively, what we see at the beginning is a full neuron that is labeled, you can see it here, but when this cell undergoes apoptosis, it breaks down, small fragments, small axonal fragments, but also these large cell bodies will form. And these basically, this is the entire cell body, and what you see dark here, this is the nucleus. So you have the production of very small axonal fragments, plus these very large. And in the lab, we mostly focus on the uptake of these large corpses, which are something like 5-6 micron wide. Okay, so now we can basically cross the two transgenic lines together, the microglia and the cell death reporter, and now we can start to make sense of the motility that I described before. Effectively, what we can see is that the brain is there are many of these corpses, and if we image a microglia, if we focus on a single microglia, we can see that the branches are directed towards these corpses. And what you can also see is that you form this large vesicle here. This is the phagosome. And then these material is brought inside the soma of the microglia, and you see this accumulation of fluorescence, which indicates the uptake of many neurons. So here is a little quantification of the speed at which these branches extend and retract. And I don't want to go too much into details, but basically this is very comparable to what has been described for the mouse. So today I would like to touch upon two little stories just to give you a flavor of what we do. Dealing with how microglia, if you allow me the expression, control their appetite. So can these cells eat everything which is here on their plate, or do they follow certain rules? Do they have to limit themselves? And then linked to this, digestion. So what happens next? And again, the crosstalk between these two things we think is really important to understand. Okay, so let's take a better look at the situation here. This is just a segmentation of the microglia. These are these green dots, and the apoptotic neurons. These are the gray dots. More than apoptotic neurons, I already mentioned, these are really the cell bodies, the corpses, the big stuff. So as you can see from this, there is really, the microglia are outnumbered by the dead cells. So each microglia is surrounded by many dead cells, okay? And so here really we are asking, okay, they have all these branches, they have all these dead neurons, can they just take them all? Or, as I mentioned, is there a limit to what they can do? Now, as we approached these questions, we realized that actually we had to improve our data acquisition. And in particular, we wanted really to look at microglia and how they scan the brain and find these neurons. And we realized that we basically needed to fulfill three very important requirements. The first one, we had to be fast because these cells are really fast. The second one is we needed to keep phototoxicity low. And in particular, when you're too fast with your microscope, let's say a confocal, you know that you are introducing photo damage. So the two things are not really going nicely together. And the third one, because this is a live process, we wanted to be as quantitative as possible in order to really extract principles on how these cells do. So we achieved this by using light sheet microscopes, also called SPIM, and both commercially available and custom made. Because basically, since you illuminate your sample with a plane of illumination, you can go a plane of light, you can go very fast indeed. It's very low in phototoxicity, and some models at least allow multi-sampling, and so being quantitative it's possible. And what we do basically is to really do we do unbiased in total imaging of the whole brain, and then via adaptive cropping we crop single cells, so we keep these cells in the center of mass, and then we can examine single microglia, both at cellular and subcellular resolution. So what did we learn from this? So first of all, by really looking at microglia and how they move and how often they interact with the dying neurons, we found that on average, a single microglia will interact, on average, a single microglia will have 10 interactions per hour with the dying neurons around it, okay? However, only half of these interactions will lead to a successful engulfment. And you can see the difference between a successful and we call it aborted engulfment right here. So a successful engulfment is characterized really by the formation of the phagosome, and then the phagosome is retracted and takes with the apoptotic cargo. This blue, it's in blue now. However, in the case of the aborted, you really have the formation of these obvious phagocytic cups, but then they somehow resolve, and the cell, the dying cell, is not engulfed. It will be engulfed in a second, third attempt. Now, interestingly, these aborted attempts, as far as we can see right now, do not depend on features of the apoptotic cells. So, for example, how old is the apoptotic cell, so since how long is there, how far, or so on. They appear quite random. Another important thing that we can draw from this is the fact that when you see a phagosome closing like this, this is an indication of a successful engulfment. And we don't always need to label the apoptotic cargo. And this is, of course, allows us to go even faster in our image acquisition. Now, there is another thing that we learned doing these, taking this in vivo imaging approach that we somehow didn't expect. Because as I told you, microglia have many arms, and they have a lot of food in front of them. However, they're very polite, and they take one neuron at the time. So they never take two dead neurons at the same time, but they always go one by one. So engulfment is always sequential. And interestingly, and again, engulfments here are marked by these asterisks, and because we can really see these phagosomes, and then we can see the transport of the phagosomes. And as you can see, one after the other. There is also high spatiotemporal correlation between two subsequent events. So what this means is that if you look at two events, which are one after each other, they are closer in time if these neurons are located close to one another relative to the microglia. However, if they are far apart relative to the microglia, it will take longer for the microglia to first take one and then take the other one. And so this really tells us that microglia are really able to polarize towards distinct dying neurons, and that intracellular mechanism must exist that allow these cells to polarize effectively towards single targets. So at this point, really, we wanted to understand, okay, how is the microglia able to say, okay, I go over there, and then I go over there, and so on. And so we approach this program knowing that, of course, other cells have to polarize too, and that often what they do is to use the centrosome, because the centrosome is an organelle that in many cells defines polarity. For example, in neuron, the position of the centrosome is define which dendrite will become the axon. Because it's a microtubule polarizing center. So we decided to actually look at the microglia centrosome, and we generated a reporter. It's a reporter that labels all centrosomes. So all these dots you see here are the centrosomes of the surrounding neurons. And here, encircled, this is the centrosome of this microglia. In many cells, the centrosome has a central position. This is why it's called centrosome. But when we looked at the centrosome of the microglia, we got a surprise. And we found basically that the centrosome moves very rapidly. And it basically moves inside the branches of the microglia, in and out. But every time the centrosome reaches the tip, which is something that you can see here, and then you can see here again, it will go down the branch. Every time it reaches the tip, that's when phagocytosis will take place. And so you can see here, quantified over many events, this is engulfment. And you can see acceleration of the centrosome towards the engulfment. You can see here, this is relative to this microglia here. Again, in red, you have the engulfment event. And as you can see, the centrosome starts moving inside the branch prior engulfment. So really, appears that the targeted movement of the centrosome predicts engulfment. In line with this, when we examine aborted versus successful events, we find that only when the event is successful, we see this movement of the centrosome towards the tip. But if we examine aborted events, the centrosome has random positions, okay? So really centrosome migration in a way provides also a mean for us to distinguish between successful and unsuccessful phagocytic events. So at this point we asked, could centrosomal position rate limit engulfment in microglia, and explain why microglia somehow only pick one branch that is going to be the successful one and are not able to engulf two neurons at the same time simply because they have one centrosome, and the centrosome can only be in one place at a time. So we decided to test this by generating microglia with two centrosomes, and you can do this by overexpressing core components of the centrosome, for example, centrin or SAC6, etc. Then you're able to make cells with more centrosomes. So if we make microglia with two centrosomes, and you can see here these two dots, these are the two centrosomes, you really allow microglia now to engulf almost double the neurons. And you can also see that these cells are able to engulf neurons at the same time. And each event has a centrosome nearby associated with. So this really tells us that this organelle is sufficient to promote engulfment, and it plays really a key role also in limiting the rate of engulfment when microglia are using their branches. So the next question is, of course, how does the centrosome know where to go? Okay, towards the tip, and why, in response to what? And again, here we were inspired by other cells that do something very similar to the microglia. And they actually, and these are T cells. So T cells establish a so-called immunological synapse with antigen-presenting cells. And it's known that in these cells, the centrosome is usually in a central posterior position. But when the T cell finds the right antigen-presenting cells, the centrosome moves towards this immunological synapse, which is the interface between the T cell and the antigen-presenting cell. And this is due to the localized accumulation of DAG, which is diacylglycerol. So this buildup of DAG, which is downstream the activation of the T cell receptors, allows the centrosome to find the right spot. It is important for the centrosome to go there because basically what the centrosome does is to bring with cytotoxic vesicles and so on. So it's an important event. So we decided to see if microglia could possibly use a conserved similar system. And what we did was to use inhibitors of, for example, PLC, which is required for this localized buildup of DIC, and then also some downstream components. In the interest of time, I'm only going to show you some data. For example, we used an analog of diacylglycerol. When we put it in when we incubated the fish embryo with this, basically what we do, we erase this localized buildup, we put it in all around the microglia. And what you can see, so there is no longer positional information or localized information. And what you can see is that, first of all, the microglia is absolutely highly motile, sending branches in all directions. But if you pay attention, the centrosome remains always very central, doesn't really adventure much into these branches. And when we quantify engulfment, this is very, very low. It's because mostly the cells have lost the knowledge of where to go and where to polarize towards. So it appears that the role of DAG signaling in promoting centrosomal reorientation, it's indeed important here. And then there is also a striking similarity and conservation between the immunological synapse and the phagocytic synapse. So what is the benefit of actually reorienting the centrosome? Why do the microglia need to reorient the centrosome? Why questions are always difficult in biology, but what we see is that bringing the centrosome in the direction of the forming phagosome really allows microglia to polarize in that direction. As you can see, the centrosome really works as a microtubule organizing center. Here we are looking at microtubule polymerization live. So you can see how really microtubules form a very strong cage around these phagosomes. These are really big vesicles that contain an entire neuronal cell body with nucleus and so on. So the idea here is that microglia need really a big force to really transport these back. Okay, so now the centrosome here is limiting the rate of engulfment, and a question, of course, that we have is limiting the rate of engulfment important for microglia? Why cannot microglia eat whatever they want? And that, of course, in some diseases would be maybe good, or will be good to know how to limit this. Now, the answer is that yes, microglia do limit their engulfment, and we really think that there are important feedback loops, negative, positive feedback loops between resolution and the ability to engulf. And I would like to quickly show you what we know about this and, you know, how we are going about this. So again, we have tracked phagocytomes. So we have looked now at how microglia actually eat. So let's keep following these phagocytomes that contain the neurons and what did we learn from this. So the first thing we discovered that was already known and shown, especially in C. elegans, in phagocytosis in C. elegans, is that, and applies also to our microglia here, is that phagocytomes, once they form, they undergo a shrinkage. So they undergo a size reduction. You can see this in the video here. It's quantified here and also here. Now, this shrinkage from 5 microns down to 2 until we can follow them basically depends on transporters that are present on the phagocytomes. One in particular, which is the SLC37A2, it's a putative transporter for glucose 6-phosphate. So basically, we are thinking that the digestion of the apoptotic cargo leads to byproducts, for example, sugars. The export of sugars from these phagocytomes is coupled with the ability to actually reduce the size. And so this process is important. I'm going to come back to this in a second. Another thing that we have learned about these phagocytomes, and that we didn't expect at all, is that these phagocytomes, they all merge into a vesicle that is always present in these microglia. And this is marked here with this red dot. So if we track vesicles, we always see them coming and going. But this one with the red dot, we can track it for hours. And what it does basically is that it fuses with all the incoming phagocytomes. And you can also see the size of this red vesicle fluctuating depending on fusion. These lines here, they mark these fusion events. And this process of fusion becomes very obvious when we have an SLC37A2 knockout, because in this case, the phagocytomes are big. And therefore, when they fuse with this collection compartment, the collection compartment keeps growing. And in fact, you know, it becomes really, really big. Okay? So we were surprised about this. And of course, at this point, we wonder, is this only microglia? Is this only fish? And so on. So we turned to raw macrophages, which are mammalian macrophages that we can culture. And we reasoned and we set up the following experiment. We fed these raw macrophages with HeLa cells that either have a green nucleus or a red nucleus. And we reasoned that if after this feeding, there is one yellow vesicle and one only, this means really that we have something similar to what we have seen in the microglia in the fish, that everything comes to one single, is collected in one single vesicle. And indeed, we saw exactly this. We wait 6 hours, 9 hours, we see everything gathering in this one spot. And we call this one spot the gastrosome, because it works a bit like a stomach, a phagocytic stomach in these cells. Now, one question we had, of course, is if this if this gastrosome is different from all the other vesicles of all the other phagocytic vesicles, so phagosomes, lysosomes, and so on. So I'm only going to show you one approach we took, which is CLEM. This is correlative light electron microscopy. So we are able to spot the gastrosome first fluorescently, and then we go and look in raw macrophages at the alta-structural level. We do the same thing in microglia, and therefore in the fish, we can compare the two vesicles in the mouse macrophages, in the fish microglia, and we really find that they have very similar characteristics. They are electrolucent, mostly surrounded by a single membrane. The gastrosome contains membrane fragments. A few other things we have done, which I'm just summarizing here briefly. The gastrosome has also distinct combinatorial molecular signature. It is LAMP1 positive, but is catapsin D negative. So it's not like your average lysosome. And as I said, the lumen appears to contain membrane fragments and lipids. So we are exploring this option that indeed it has something to do with lipid metabolism in these phagocytic cells. So we are still investigating the nature of the gastrosome. We still don't know exactly what it does, but one very important thing we know is that this vesicle is really constitute an important bottleneck in phagocytic cells. So I show you just a little experiment. This is my last slide on this. We can really make microglia in the fish with a very large gastrosome by increasing the level of cell death. We use cantotecin, is a drug that kills highly proliferating cells. So in this case, neurons. As the microglia starts to engulf more, the gastrosome now, faster, let's say, the gastrosome now cannot keep up and really becomes larger and larger. Now, when you have cells that have such a large gastrosome, what we have learned is that these cells, for example, are unable to move towards an injury. So normally we can kill lots of neurons and the microglia will migrate there. But if you have cells with such a large gastrosome, they don't move, not surprisingly, considering the size of these cells. But they are also phagocytosing dead neurons much less. So a little bit like the Omer here, in a way, they are, bloated, and even although they have eaten a lot, they are sort of done with it. And of course, right here, we are very intrigued by the possibility of this being a way to really manipulate these cells, right? You, in certain contexts, in certain disease, you want to reduce their, appetite as much as possible. And a way to do it is really by getting, acting at the level of their stomach. So making these cells think that you had too much, and so you should stop now. And this is, avenues that we are exploring. Okay, then, finally, just to summarize a little bit. So yes, for us, the gastrosome might be an important epicenter in these cells. What is its function? We are still investigating this. And we are intrigued by the fact that often lysosomes are considered the metabolic centers or nutrient sensing organelles in a cell. But this, the existence of such a structure in phagocytic cells will shift a little bit the attention from the lysosomes to this compartment. And we are still working on this, many questions. So stay tuned for the future, hopefully. So I would really like to thank at the end the people that did the work that I presented. So the work on the centrosome is from Katrin Müller, who was a former PhD student in the lab. She's now a postdoc in Iceland, as you can see from the volcano at the back, I think. And then the work on the gastrosome was initiated by Ambra Villani and Jürgen Benjaminsen. Ambra is in the lab and is working hard still on the gastrosome, together with Joanna Zareba, who has just defended her PhD. And thanks to, of course, all the others that contribute with ideas, discussions, and so on. And thank you all also for your attention. And I'm very happy to take questions. Thank you.
Thank you, Francesca. It was a very nice talk. And it's so nice that you already have a lot of questions. And I have to say, at some point, we were like 190 people. So, yay!
It's because it's the first one. I hope.
No, it's the second.
It's the second, sorry.
So Marta Pereira will introduce the Q&A. She will handle them. So ahead, Marta.
So just a little reminder, you can write your name in the chat and we will unmute you, or you can write the question and I will read it. So as Amanda said, beautiful talk. We already have a lot of questions in the chat. So the first one is from Ejo and it says, is the gastrosome positive for lipofunction?
Lipofusion.
Lipofusion, you mean? Yeah.
Yes.
This is something we didn't check yet. It's a very good point, and we have, of course, it's in our list, but I can't really say for sure. Don't have conclusive data on this. Yeah.
Then the next one is from Chen, and it says, in terms of the successful aborted engulfment, could it be the apoptotic neurons releasing different signals to microglia that also determine the date of phagocytosis?
Sorry, I needed to rehear a second. I got distracted.
If it could be the apoptotic neurons releasing different signals to microglia that also determine the date of phagocytosis.
Different signals, so the molecular nature of these signals will be different.
From the neurons.
From the neurons. So for sure, one thing we know is that the engulfment of apoptotic cells at the molecular level is really a jungle, in the sense that lots of signals are known to attract phagocytes. So it's well possible that different signals are going out to alert the phagocytes, and the phagocytes have different receptors. It's possible, and this might lead to a different outcome. However, we don't really have evidence for this in our system. As far as we know, the attraction is mediated by nucleotide release, appears to be mediated by nucleotide release. But yeah, it's possible that there are other mechanisms, but we don't know for sure. Yeah.
Okay, then Agnes says, beautiful talk, thank you. Do you think that the gastrosomes also apply to microglia phagocyting smaller cargos, such as the spines?
Yeah, this is a very good point. Yeah, I mean, maybe I guess synapses or smaller things. I don't know, honestly. We don't have evidence for this, so I can't tell for sure. But it's possible that it remains an important vesicle that will be there even if microglia are not engulfing big cargos. It might change its size, so it might be smaller. And this is something that we are currently investigating, if the gastrosome is always there, or if it appears when cells are highly phagocytic and when they are phagocytosing big targets. So something that, interesting point.
Carmen says, nice talk, beautiful videos. Is the phagosome gastrosome similar independently of the substrate? So, for example, if it is different, if the substrate is sugar-rich or lipid-rich?
So this is a very interesting point. Our idea at the moment, and this is something that we are looking into, is that not everything might end up in the gastrosome. And for example, we see that SLC is localized already on phagosomes. So SLC is responsible for export of a sugar. So our idea is that different byproducts of digestion will exit the phagocytic pathway, let's say, at different point. So sugar might, go first, and then who knows about nucleotides? We don't know exactly. But then, some byproducts will end up in the gastrosome. Maybe lipids, as I said, because we see membrane and we see some evidence for lipids being there. You know, at the moment we think that is not a generic storage, but that, you know, things might differentially behave along the phagocytic pathway. Yeah.
Mm-hmm. Then Sophia says, amazing talk. Do you think that lipid accumulating microglia has huge gastrosome and responsible for its phenotype?
Lipid accumulating microglia. Oh, yes. In many diseases, there is evidence for lipid droplets being present in microglia and accumulation of lipid microglia. Yeah, I don't know, but yeah, would love to find a link there, of course, if that is the gastrosome. However, I also think that there is more than just the gastrosome. We know of lipid droplets, for example. This would be other compartments, but yeah, we love to see the gastrosome involved there. Yeah,
sure. Then Frances said, fascinating talk. For an immunohistochemistry experiment, what would be the best marker to distinguish between lysosome and the gastrosome?
We don't have a marker, and that's our problem. Yeah, we don't have a marker yet. So we are really keen on doing, we have started already proteomics and lipidomics and so on. But, you know, the problem is, and we have to be realistic here, the phagocytic pathway is a very promiscuous pathway where every vesicle fuses with everything else. So even when it comes to LAMP1, RAB7, and RAB5, which are assigned to the different vesicle types, in reality, you find them a little bit everywhere. So it might be possible at some point for us to identify a marker, but it's also possible that this might be a little bit difficult, simply because really these vesicles, they fuse with one another constantly. So we know that the gastrosome is different from the lysosomes because it's bigger, the ultrastructure is different, and so on. But we don't have something that is localized either inside, or it is a membrane that is unique. And yeah, we hope to find it, of course. Mm-hmm. It will make our life easier. Yeah.
Yes. Then Alexandra asked, great talk, thanks. You mentioned the microglia engulfing both dying and dead neuronal cells. Were you able to tell the difference between the two? And also, were there any examples of the microglia engulfing healthy neurons?
Someone from Guy Brown or okay, yes. Yes, I'm very not very precise with. I've not been very precise with my language. Dying dead cells. Often I'm referring mostly to apoptotic. Although we have done in the past experiments using lasers and killing neurons, so necrotic cell death, and these are also engulfed. Didn't really look at the role of the gastrosome there really. Live neurons, we have not looked into this at all, so I don't know. I would imagine a lot of interactions, but engulfment of a live neurons. You know, often you then doubt if your marker is there or not, so it's a bit difficult. Yeah, but it's a very good point. We should look into this.
Mm-hmm. And then Petros says, many thanks for the beautiful talk. Are the same principles of microglia engulfment of dead neurons follow in phagocytosis of bacteria?
There are differences between the phagocytosis of apoptotic cells, that is also known as efferocytosis, and phagocytosis of bacteria. Different receptors and often different mechanism. For example, in the case of the microglia, the membrane is, so you really have the formation of a phagocytic cup where you have membrane protrusion and changes forming these phagosomes, and then is retracting. often bacteria simply sink inside. Now we have collaborations with people to see indeed if exposure to bacteria will also lead to a role for the gastrosome there. We don't know yet. We don't know yet.
Then Victor Tapia says, great talk, Francesca. Will an annexing reporter label any apoptotic cell in the brain tissue? How do you recommend to follow apoptosis of specific cell types? For example, does a neuron promoter cytosolic GFP, for example, line will keep the fluorescence when cells undergo apoptosis?
No, so basically our line really allows us to pretty much only see the cells that undergo apoptosis, and as I said, a lot of the fragments there, because really there is a build-up outside of the cell, so which becomes really decorated with this marker. It will label any cell, yes, of course, because basically it's secreted and then diffuses and then, label apoptotic cells. So in my view, really to label a specific cell type, one would need to use maybe a caspase-3 reporter, and those are there are caspase-3 reporters that will report on the activity of caspase-3 in one specific cell type, and often that will lead to a labeling that will remain, that will be there until that cell is engulfed. We didn't use this in our system simply because it didn't work in the past, and there is no much cytoplasm in these neurons there, so it didn't look really ideal for us, and we were lucky with our reporters, so we kept using that. But that would be a way to go, in my view.
Keijin Su says, interesting talk. Do you know what molecular cues are responsible for the formation of the gastrosomes? And would you expect a change in the size or phenotype of gastrosomes in dams as compared to other microglia?
Okay, with molecular cues are responsible for the size increase of the gastrosome? No, I'm afraid for many of these questions, my answers are a little bit always like, no, we don't know. I was like, no, we don't know.
Okay.
Lots to we can conclude, I think.
So it says, thank you, great talk. Have you planned to study this further in human samples or using human iPS-derived microglia? For example, during development of microglia containing brain organoids?
Yeah, it's a very good point. Of course, we are very interested. Yes, something to do for sure.
Yes.
A very good point. Yes.
So let's go with the last two questions. The first one is from Alexia. Thanks for the great talk. Have you thought about investigating whether neuronal activity modulates the speed of this phagocytosis of dead neurons? Maybe by correlating the size of the gastrosome with calcium imaging data of surrounding neurons.
Mm-hmm. Yes, that's a very good suggestion. We do have calcium reporters, and we have indeed, this is one of our dream to make sense of the dynamics of the microglia also in relation to surrounding neuronal activity. It requires really fast imaging, and it's not trivial something not trivial to do. Microglia are really complex cells in the 3D space, and the neuronal activity is a very fast thing happening. So if you want resolution, temporal and spatial resolution, it's a bit of a challenge also for the zebrafish system. So love to do that, of course.
Okay, and the last one from Guy Brown, great talk and beautiful images, and he asked whether you ever see neuronal corpses go into the microglia but then come out.
Basically what we see are these aborted events, right? So, but if the phagosome closes and then is transported in, no, I don't think that is going to resolve in any way that the neurons will then be released out. Of course, right? If we watch this process happening, we do see that there are these events where these phagocytic cups are almost closed, but then they open again, and then the neuron is left there, and then after it will be engulfed, maybe there is another attempt and so on. So our live imaging approach shows this, and shows that when the phagosome is closed and is transporting towards the cell body, this is it. This is engulfment. I can exclude, of course, that something might happen, but we have never seen that. Yeah.
Great. I think this is it for the Q&A.
Well, Francesca, you really endured a lot of questions.
Yeah, but I feel terrible.
No, it was amazing.
I was like,
think
oh, great.
it was a great interaction with the audience.
More people.
And you were exactly on time, you know, it's exactly 6 o'clock, so perfect timing.
Gosh, you see Swiss.
Yeah, so people, maybe you can turn on your cameras and say thank you and goodbye to Francesca. You can turn on your microphones, say thank you. She gave a great talk.
Thank you.
Thank you.
Thank you.
Bye, people. Thanks, everybody. And just remember, this will be uploaded in our channel, so we will post it online for you to share.
The next one will be on December 4th.
December 4th, yes. Who's the speaker? Renzo Mancuso.
Oh,
yes.
wow.
Yes.
That's great.
So thank you, everybody.
Thank you, Francesca.
Thank you so much.
Bye.
Thank you so much again for the invitation. Have a nice evening.
Bye.
Bye.
Bye.